Chapter 6 covers metabolism, which is one of the 7 properties of life, because all things must take in and use energy. The study of metabolism accounts for all of these activities. In this video, though, we're only going to cover the basics. We're going to explore specific reactions in the next 2 chapters. Bioenergetics is a branch of biology in which the building and breaking of molecules and stepwise chemical reactions is studied and explored. All chemical reactions in cells that use and release energy are considered a part of the cell's metabolism. Most life forms on Earth get their energy from the sun. Plants use photosynthesis to capture sunlight, and herbivores eat those plants to obtain their energy. Carnivores eat herbivores, and decomposers digest plant and animal matter. The metabolism of sugar is a classical example of the energy reactions in a cell. The breakdown of sugar is seen in the top equation, which is what animals do in order to release usable energy into their cells. This process is called cellular respiration. The second equation is the creation of sugar, which is what plants regularly do. The process is called photosynthesis. The making and breaking down of sugars illustrates 2 different types of metabolic pathways. Understanding how organisms metabolize energy allows us to study their evolutionary history. This tree shows the evolution of various branches of life. The vertical dimension is time. The early life forms in blue use something called anaerobic metabolism to maintain energy from their surroundings. Aerobic metabolism evolved later. We label reactions as either anabolic or catabolic in nature. Anabolic pathways are those that require energy to synthesize larger molecules. Catabolic pathways are those that generate energy by breaking down larger molecules. Both types of pathways are required for maintaining a cell's energy balance. When studying metabolism, you must talk about energy, which is technically defined as the ability to do work, and there are different types of energy. Kinetic energy is the energy associated with objects in motion. That would be the water flowing in the stream on the right. Potential energy is the energy associated with the ability to do work or move. That would be the water behind the dam. That has a lot of potential. Potential energy refers not only to position, like the water behind the dam, but also to energy that is locked in chemical bonds. Therefore, chemical energy is technically a type of potential energy. The release of energy is brought about by breaking the molecular bonds, like those within fuels. We measure the energy in a system by looking at the changes in free energy. The change in free energy can be calculated for any system that undergoes such a change, such as a chemical reaction. To calculate delta G, or the change in free energy, all you have to do is subtract the amount of energy lost to entropy from the total energy change in the system. We'll talk about entropy in just a second. This total energy change in the system is called the enthalpy, and it's denoted as that delta H, or that triangle H. The formula for calculating delta G is as follows, where the symbol T refers to the absolute temperature in Kelvin. Kelvin is just something we use in science to be more exact. You calculate Kelvin by taking the temperature in Celsius and just adding 273. The standard free energy change of a chemical reaction is expressed as the amount of energy per mole of the reaction product under standard pH, temperature, and pressure conditions. When we say standard pH, temperature, and pressure, we're talking about a pH of 7, a temperature of 25 degrees Celsius, and about 1 atmosphere. It's important to note that cellular conditions vary considerably from these standard conditions, so the standard calculated delta G values for biological reactions will be different inside the cell. We'll see these values denoted on various things when we talk about cellular respiration and photosynthesis. I would just like you to know where they come from. If energy is released, the results of that equation we just discussed are negative. The reaction is considered then an exergonic reaction. If energy is being taken in order to be stored, we say that the net result of that equation we've studied is positive. The reaction is therefore considered endergonic. Shown here are examples of endergonic processes and some exergonic processes. These include things like compost pile decomposing, a chick hatching from a fertilized egg, sand art being destroyed, or in a ball rolling down a hill. See if you can attach endergonic and exergonic to each of these examples. Our bodies, thankfully, aren't in great constant flux. There is some chemical equilibrium to our systems. Many chemical reactions are reversible, though, so bonds can be made, broken, and then remade. Reactants in a closed system will occur until this nice equilibrium is reached. Energy must be added to a system to push the equilibrium in one direction and keep it there or allow it to bounce back and forth. A constant supply of energy is supplied to our planet from sunlight. It's important to note that I said reactions in a closed system will occur until this equilibrium is reached. The cells that we study, the cells in our body and in our natural world, are not a closed system unless they're dead and preserved. All of these general reactions have a net release of energy, but still require some energy in the beginning. The small amount of energy input necessary for all chemical reactions to occur is called the activation energy, and we abbreviate it with a capital E and a little a. This tiny energy requirement is because the bonds of molecules must be contorted into a new shape in order to break them. This contorted state is called the transition state of a molecule. It has a lot of energy and it's very unstable. For this reason, reactant molecules don't last as long in their transition state, but quickly proceed to the next steps in the chemical reaction. That little bit of energy can be overcome a couple of ways. Heat energy can give you the energy of activation that you require. Heat speeds up the motions of molecules, which increases the frequency and force in which they collide. This can allow their atoms to move and bend slightly, and it helps them reach their transition state a little bit faster. Heating up a system will cause chemical reactions within that system to react more frequently. If you increase the pressure in a system, it has a very similar effect. Once reactants have absorbed enough energy from heat and they reach their transition state, whatever reaction they were kind of heading towards, it's going to proceed and the reaction will take place. The higher the activation energy, the slower the chemical reaction will be. It takes a long time to get over that activation energy hump so you can get the ball rolling down the hill and continue on with your reaction. The activation energy requirements for most cellular reactions is too high for heat energy to overcome it at efficient rates. In other words, in order for important cellular reactions to occur at appreciable rates, the number of reactions per minute, their activation energies must be lowered. The lowering of activation energies is called a catalyst. It's a very good thing as far as living cells are concerned that these catalysts exist. They allow us to undergo more reactions in a given day and therefore use and consume more energy and do more things. If cellular temperatures alone provided enough heat for exergonic reactions to overcome activation barriers, essential components of cells would actually disintegrate. Having too much activity happening is actually quite dangerous. Our proteins, DNA and RNA, they have a lot of bonds and therefore store a lot of energy, could essentially fall apart simply because we got too hot. There are laws in physics that determine how energy is transferred and how it exists in the natural world. The study of energy is referred to as thermodynamics. Let's talk about the two laws of thermodynamics and we'll see how all these energy equations came about and why we need to worry about things like heat. The first law states that the total amount of energy in the universe is constant. There has been and will always be the same amount of energy. Energy can be transferred from one state to another, and this happens all the time, but energy can neither be created or destroyed. There is no completely efficient energy transfers in the universe. Some energy is lost to a form that is too unstable to be recaptured. In thermodynamics, we talk about heat energy. So we're back to heat. Heat energy is energy that's transferred from one system to another as a result of some sort of reaction, but that energy can't do any work. This also can be thought of in terms of entropy. Entropy is the increasing state of disorder in all things. We have to put energy into systems repeatedly in order to keep them organized. Think about it like having to actively clean your kitchen so your sink won't smell, or you actively have to do laundry to keep your room from being a mess. Things are constantly happening in those systems. We have to input energy to keep entropy in check. Within cells, energy comes from a molecule called adenosine triphosphate, or ATP. ATP is an adenosine bound to 3 phosphate groups. The adenosine itself is a nucleoside consisting of a nitrogenous base called adenine and a 5-carbon sugar called ribose. The 3 phosphate groups are labeled alpha, beta, and gamma. Not all of the bonds within this molecule exist in a particularly high energy state, but some do. Both of the bonds that link the phosphates are equally high energy, and when broken, they release sufficient energy to power a variety of cellular reactions and processes. The reason these bonds are considered high energy is because the products of the bond breaking, ADP and organic phosphate, have a considerably lower free energy than the reactants. It takes ATP and a water molecule to actually split these up. And because the reactions take place with the use of the water molecule, this is considered a hydrolysis reaction, which we know all about from our studies in biological macromolecule making. Like most chemical reactions, the hydrolysis of ATP to ADP is reversible. The reverse reaction generates just a new ATP from ADP and PI. Indeed, cells rely on the regeneration of ATP just as people rely on the regeneration of spent money through some sort of income, hopefully. Since ATP hydrolysis releases energy, ATP regeneration must require an input of free energy. The formation of ATP is expressed in the equation seen here. We are going to talk ATP into the ground when we do our chapter on cellular respiration. So, about that ATP making. The delta G for the hydrolysis of 1 mole of ATP into ADP is about negative 7.3 kilocalories per mole. This is under true standard conditions. So in a perfect world, that's how much energy is released into the system when you break up an ATP and those two individual pieces. ATP is an extraordinarily unstable molecule. If you don't use it right away, it's going to fall apart completely on its own, and the free energy is just lost into a system as heat. Heat comes back again. Cells couple exergonic reactions of ATP hydrolysis with endergonic reactions. So if you're making ATP, you're almost always immediately using it right away. That's that coupling it with another reaction in order to ensure that you didn't just waste your time making an ATP that you know is just going to fall apart. A large percentage of a cell's ATP is spent powering this pump, which is hopefully familiar, because cellular processes bring in a great deal of sodium into a cell, and potassium ends up getting pumped out. The pump works constantly to stabilize cellular concentrations of sodium and potassium. In order for the pump to turn one cycle, so getting rid of 3 sodiums and bringing in 2 potassiums, 1 molecule of ATP must be hydrolyzed or broken up using water. Once that ATP is split, the gamma phosphate doesn't just walk away, it actually gets transferred, it gets put on that little protein pump. The process of a phosphate group binding to a molecule is called phosphorylation. With the addition of the phosphate, the protein pump has the ability to change shape. We learned about that shape changing. It will help with the exchange within the pump of pushing the sodium out and bringing the potassium in. There are some special molecules that allow less energy to be used to do all these various reactions. The special molecules that catalyze biochemical reactions are called enzymes. Almost all enzymes are proteins, and they're made up of chains of amino acids. They perform critical tasks like lowering the activation energies of chemical reactions inside of a cell. Enzymes do this by binding to the reactant molecules and holding them in such a way to make chemical bond breaking and forming processes take place more readily. They only reduce the activation required to reach the transition state. The chemical reactants to which enzymes bond are called the enzyme substrates. The location within the enzyme where the substrate is actually doing its bonding is called the active site. Since enzymes are often proteins, there's generally a unique combination of amino acid residues, the little R groups we learned about, within each of those active sites. Each residue is characterized by different properties. The substrate binds with an enzyme on the active site, and the special binding is called an induced fit model. According to the induced fit model, both the enzyme and the substrate undergo dynamic conformational changes upon bonding. The enzyme contorts to a substrate into a transition state, and therefore increasing the rate of reaction. You might have heard about a lock and key mechanism when it comes to enzymes and substrates. I put the lock and key and the induced fit model together in my mind by thinking about hugging a friend. You hold your arms out, one's a little bit lower than the other, and your friend does that too. And you come together in your hug, but you don't stay in that awkward position. If it's a good friend, you probably wrap your arms around them and pat them on the back. You're coming together because you have a specific shape and they have a specific shape, but then you induce the fit and get a better hug. Enzymes do that too. Enzyme regulation is very important to the health and function of a cell's metabolism. Enzymes can either be promoted or inhibited in their actions, and when you're talking about that inhibition, it can either be competitive or non-competitive. Competitive and non-competitive inhibition affect the reaction rate differently. Competitive inhibitors affect the initial rate, but do not affect the maximal rate, whereas non-competitive inhibitors affect the maximal rate. Check out your book for further explanation of those concepts. Some inhibitor molecules bind to the enzymes in a location where their binding induces a conformational change that reduces the affinity for the enzymes for their binding sites. This type of inhibition is called allosteric inhibition. Most allosterically regulated enzymes are made up of more than one polypeptide. This means that they have more than one protein subunit. When an allosteric inhibitor binds to an enzyme, all the active sites on the protein subunits are changed slightly, such that they bind with their substances less efficiently. This is an example of competitive inhibition. There are allosteric activators as well as inhibitors. Allosteric activators bind to the locations on an enzyme such that a way that the active site actually has more affinity for the substrates than it did before. This is obviously not competition at all. Few enzymes work entirely on their own. Most need cofactors or coenzymes to assist them in their binding and functioning. Cofactors are just inorganic helpers, while coenzymes are organic ones. The abundance of these molecules helps to keep enzymes in check. If we don't have the right vitamins and minerals in our bodies for processes to work out their pathways completely, there's no point in wasting energy to begin them, so we just stop them at the get-go. Enzymes are kept in certain components for their safekeeping. It's a really bad idea to have molecules that are reactive just floating around in your cells without any form of protection. The most relevant sources of enzyme regulatory molecules, at least when you're talking about cellular metabolism, are actually the products of the cellular metabolic reactions themselves. In the most efficient and elegant way, cells have evolved to use the products of their own reactions for feedback inhibition of enzyme activity. Feedback inhibition involves the use of a reaction product to regulate its own further production. The cell responds to the abundance of specific products by slowing down production during anabolic or catabolic reactions. Such reactant products may inhibit enzymes that catalyze their production through the mechanisms described in this picture. We'll talk about all of these enzymes and their pathways a whole lot more in class. I understand that this video seems a bit vague as of right now. If you read your chapter, I think a lot of this enzyme material is going to make a little bit more sense. We'll explore it in class. Try to answer the questions at the end of each section and complete your learner homework, or at least take a peek so you know what to expect.